Hi, my name is Carlos Hernandez, and I'm from Ciudad Juárez, and I'm from the Biomedical Engineering Program at the Universidad Autónoma de Ciudad Juárez. Today, I will present our work entitled Cell Viability Evaluation of Tubular Nanoparticles in Collagen System for Wound Healing Application. This project focuses on the development and preliminary evaluation of a hybrid biomaterial composed of silver nanoparticles incorporated into a type 1 collagen matrix with potential application in wound healing. Wound healing is a complex biological process that can be negatively affected by factors such as infection, inflammation, and delayed tissue regeneration. For this reason, there is an increasing interest in developing biomaterials that can simultaneously support tissue regeneration and help reduce the risk of infection. In this project, we focus on two materials with complementary properties. The first one is collagen. Collagen is one of the main components of the extracellular matrix and is widely used in the tissue engineering because of its biocompatibility and its ability to promote cellular adhesion and tissue repair. The second component is silver nanoparticles. The silver nanoparticles are particularly interesting because of their well-known antimicrobial and anti-inflammatory properties. Therefore, our approach was to combine both components into a single system. Collagen provides the biological matrix, while silver nanoparticles provide additional functional properties. Our objective, the general objective of this study was to determine the cell viability of the system based on silver nanoparticles and collagen produced using a green synthesis approach for potential wound healing application. To achieve this objective, we first synthesized the silver nanoparticles, characterized their physicochemical properties, incorporated them into a type 1 collagen at different concentration, and finally, evaluate the biological response of the result system using fibroblast cells. The methodology can be summarized in 6 main stages, as shown in the diagram of the poster. First, silver nanoparticles were produced using a green synthesis method based on the method proposed by Wang and collaborators. In this process, starch was used as both as reducing and stabilizing agent. This approach was selected as a more environmental friendly alternative to conventional nanoparticles synthesis method. After synthesis, the silver nanoparticles were characterized using UV visible spectroscopy, FTIR spectroscopy, and dynamic light scattering, or DLS. The nanoparticles were then incorporated into a type 1 collagen matrix at different concentration. The resulting collagen silver nanoparticle system were characterized by FTIR in scanning electron microscopy, or SEM, to evaluate the interaction and distribution of the particles within the collagen matrix. Finally, the biological response was evaluated using fibroblasts. Cell viability was quantitatively evaluated through the MTT assay and qualitatively supported by a live depth fluorescence assay. Now, moving to the results. As first, the formation of the silver nanoparticles was confirmed by the UV visible spectroscopy. As shown on the figure 2, the spectrum presents an absorption band approximately of 406 nanometers. This band corresponds to the surface plasma resonator associated with silver nanoparticles and therefore support their success formation. The nanoparticles were also analyzed by FTIR. The spectrum shows signals associated with functional groups related to starch, supporting its role into the reduction and stabilization of the nanoparticles using the green synthesis process. The dynamic light scattering was then used to evaluate the particles in suspension. The DLS analysis showed an unimodal distribution with an average hydrodynamic diameter of approximately 1,408 nanometers and a polydispersity index of 0.009. Although this hydrodynamic diameter is considerably large than the typical size expected for individual metallic nanoparticles, it is important to clarify that DLS measures the hydrodynamic behavior of the particles in suspension. Therefore, this result may be associated with aggregation and interaction between the nanoparticles and the dispersion medium rather than the size of individual silver nanoparticles. After the characterization, the silver nanoparticles were incorporated into the collagen matrix. The same micrograph shown in figure 6 allow us to compare the collagen control, this one, with the different collagen and silver nanoparticle systems. The collagen control shows its characteristic morphology, while the system contained silver nanoparticles show the presence of particles within the matrix, as you can see in the figure. Importantly, the incorporation of the nanoparticles did not produce major alteration in the overall morphology of the collagen. In addition, the same analysis suggests a more homogeneous nanoparticle distribution at intermediate concentration, particularly around 1 and 2% of silver nanoparticles. As you can see, it's more homogeneous in these images. The FTIR spectra of the collagen in silver nanoparticle system also preserved the characteristic bands associated with collagen. This should suggest that the incorporation of silver nanoparticles did not significantly alter the chemical structure of the collagen matrix. One of the most important parts of this study was the biological evaluation. Sorry for that. As shown in the figure 7, the MTT demonstrate high fibroblast viability for all evaluated concentrations. The approximate relative viability values were 98.9% of the 0.5% of silver nanoparticles, 96% of the 1%, 93% of the 2% of silver nanoparticles, and 91% of the 3% of silver nanoparticles. Therefore, even though a slight concentration depends, reduction in the cell viability was observed in all the evaluated systems, maintaining high viability. Importantly, all concentration remain above the 70% viability expectation established by the ISO 10993 for in vitro cytotoxicity evaluation. Additionally, not statistically significant differences were observed among the evaluated groups. These results were supported by the live and dead fluorescence assays shown in the figure 8. As you can see, the images show a clear predominance of viable cells across the different conditions, with morphology and cell adhesion compared to the control. Together, the MTT and live and dead cell results indicate that the incorporation of silver nanoparticles into the collagen matrix did not produce significant cytotoxic effects under the condition evaluated in this study. In conclusion, we successfully obtained silver nanoparticles through a green synthesis method using starch as a reducing and stabilizing agent and incorporate them into a type 1 collagen matrix. The physicochemical characterization confirmed that the formation of the silver nanoparticles and their incorporation into the collagen system without major structural alteration. Most importantly, the biological evaluation demonstrates high fibroblast viability across all evaluated concentrations, where the physicochemical and biological results are considered together. The 1 to 2% of silver nanoparticles range appears particularly interesting because SEM suggests a more homogeneous distribution of nanoparticles while cell viability remains high. However, these results should be considered a preliminary evaluation. Additionally, studies, including antimicrobial testing and further biological characterizations, should be necessary to confirm the functional testing and further biological characterization will be necessary to confirm the functional performance on the system for wound healing application. Overall, all results suggest that collagen and silver nanoparticle systems have promising physicochemical and biological properties that may provide a basis for the future development of biomaterials for wound healing and tissue engineering application. Thank you for your attention.